Feeling comfortable? You're not struggling with disaccharides anymore? We'll talk about modifications just one brief second again, but mostly I just want you to remember that that happens along with branching when we get into our complexity of the sugar code that we're about to encounter. So, all right. So these sometimes get confused with where we're headed. What is the distinguishing feature of a glycosaminoglycan? Tell your neighbor what those are. They're bolded. This is a tricky memory. What is the distinguishing features of glycosaminoglycans?
Glucosamine or lactose?
Oh, you're right. They're all.
Okay. After you've imprinted that, remember that all of these were examples of that very thing. And we ended talking about heparin, and heparin was the most electronegative. All right, so glycoconjugates, glycoproteins. Half of mammalian proteins are these. That's a lot. So half of your proteins have sugars attached to them. They're attached via the anomeric carbon. Protein. This, when it's with viruses, allows them to evade the immune system. Only some bacteria glycosylate a very few of their proteins. That means bacteria are easy targets for your immune system to recognize, right? They're not so good, but viruses not so much, right? Proteoglycans, glycosaminoproteins, anomeric carbon attached to a serine, hydroxyl, or an amine. And I should actually edit that serine 3-amine, right? All right. Well, we feel pretty good about all that. Is he allowed to do that? Yes, I am. Okay. There's also something about aggrecan cores. We'll talk about those. So we just mentioned this. Glycoproteins, simply said, are what's the majority?
Protein.
Protein. And then if it's a proteoglycan, more sugar. Okay, that seems reasonable. Here is a proteoglycan. Okay, that means there's more what? Sugar than protein. There's this core protein in the center of this, and these are hyaluron. These look like little, that looks like a caterpillar. I don't know, but covers joints of your articular cartilage, holds lots of water. We've already seen hyaluron involved in that before. Glycolipids, the same game we just played with proteoglycans and glycoproteins. Now I can call them glycolipids or lipopolysaccharides, fancy names, okay? All right. Blood groups. Have you ever wondered about your blood groups? There's two parts of it. We'll talk about lipids in the next unit, okay? And so this topic will come up again. But in order to recognize your blood group, it's actually several pieces of this. On your membrane of your red blood cells lies these beautiful sphingolipids with fatty acids, okay? And then attached right here is a series of what? What are these things? Sugars. These are sugars, okay? This one, by the way, is which type of sugar?
Good.
An L sugar.
Yeah.
You use them? Yes, you do. So you can't go on, I'm exclusively all about D's, right? But very few. Now, how many of you know what your blood type is? Hopefully most of you. It's a good thing to figure out. What's an easy way to figure it out? Go donate, right? Good choice. My dad loved the fact that he worked at a hospital, so it was really convenient. So he loved the fact that he had over a 55-gallon drum by the time he ended his career that he'd given him blood. It's a lot of blood.
Good for him. That's a lot.
It's a lot of blood.
Good man.
He feels like he's doing the right thing. I felt like he'd go anemic, but that's all right. So, what makes O the universal, right? Versus A and B. Maybe you have A, B. What makes O different?
0.
Yeah.
It's got the same core, but it doesn't have that extra substituent that A and B have.
Okay, so
Or that extra
specifically, if I'm A, what do I have? N-acetylgalactose. Okay, and then if I'm talking here, just galactose. So actually, those are pretty similar themselves. The N-acetyl group, which is primarily going to go on which carbon? Number 2. That's right. So these are very, very similar. So we tried, knowing that it was the sugars that were the recognition, that it would trigger your immune system. So if you're AB, you don't respond to either of these. It's just fine because you're making it, you're fine. If you're O, you don't respond. You don't have any problem. But if I give you this one or this one, it's a problem, okay? If I am A and I give you this one, problem. A and you give me this one, it's okay, right? B, it's a problem if you give me A, or no problem with O. What is it that we're saying the problem is? What's causing the problem? Madeline?
The difference in sugars you have, because the O has like the common 4 sugars that the other 2 have, and it doesn't have that long end that differentiates it.
But what is causing the problem?
So your immune system, your innate immune system is constantly like interacting with the cells of your body, and if it recognizes something that's called an antigen, which is to say it doesn't have the appropriate carbohydrate sequence, then it'll attack it.
Okay, so it's going to attack it with your immune system because it doesn't have the right sugar code. I'm using that word loosely right now because you don't know what it means yet, but sugar code. That's what it is. It doesn't have the right one, it decides to attack it. Is that potentially lethal?
Yes.
Yeah, actually it really is. Okay, so somebody went to work and they thought, well, what if we just cut them all off? What if I cut all the sugars off of your red blood cells? Would that cause an immune response? And the answer is no.
No.
Oh, really?
No immune response. Fantastic. But how long did it stay in their blood? How long did those red blood cells stay? Normally
Only
3
months, 4 months?
Normally 3 months, and how long when we cut all the sugars off?
2 weeks.
Less.
1 day.
Yeah, essentially it just filtered and removed all the red blood cells. They had fully capable of carrying oxygen, and it just ejected the cells. It just got rid of them. So being a donor with no sugars is no good to you. We can't just make a universal donor. We actually need those sugars or else they're being removed. That's kind of, you understand why they try that, right?
Yeah.
Because then I could make anybody a universal donor. Just cut off those sugars. But it didn't work. All right, so did I see another question? You good? All right.
Do we know why yet?
Nope.
Okay.
Observation. Yeah. So here's some other glycolipids. Not important that you know all these things, but lipopolysaccharides, you already talked about those. Those were external on what?
Your cells.
Your cells? Your cells? Yes. The ones I was meaning to talk about was those found in area as well, right?
Oh, yeah.
Okay. The sugar code itself, if you have the old version of my slides, excuse me for editing them because it finally bugged me enough that I went back and fixed it. I inherited some things of this slide and then I got rid of other things. This is on the write-on, and I'll tell you what's important. It's not the numbers, so that's kind of irrelevant, but I'm going to show you how you properly calculate the numbers, which is what's different from the old version versus the new version. If we compare a hexanucleotide, what is a hexanucleotide? It's made out of nucleic acids, and it's got how many of them? 6 of them. If I compare a hexanucleotide, I actually can end up with, ironically, 6 factorial divided by 6 minus 4. What's the 4 coming from? The 4 options of the nucleotides that are there, and I end up with 360 possibilities. Wow, that's kind of crazy. So that sounds like I can create an awesome genetic code, right? Yeah, it's just a permutations calculation.
So what was the R in that?
Yeah, so R is what? The number of possibilities. So number in my chain, and then R is just, I got 4 nucleic acids I'm using my DNA, right? Okay. So these are what we're talking about. They're only linked in one way. Now we'll talk about this in this unit, but I 5' to 3' linkage, it's condensation reaction. Only one option to link them. If I did a hexapeptide, 6 amino acids, but I really had 20 options, I could have done D or L, but we're going to ignore that because we almost know that we all will use just what for amino acids? L, that's right. But there's only one linkage that we use for them, which is true until I tell you otherwise. There actually is other linkages, but we don't use them for protein expression. We use them in small peptides. But they're the carboxylate and amino group. We're doing N-terminus to C-terminus. That gives me 2.8 times 10 to the 7th permutations. I'm not showing the math, but what are we doing?
It's the same.
Yeah, yep, same sort of game. All right. So, turns out that there's trillions of hexameric oligosaccharides. Now, if you look at the other one and it says 14 common hexos, that's actually true if I'm attaching it to a protein. If I'm attaching it to a just regular sugar, how many ways can I attach them? 20 common hexoses, that means 6 membered rings. How many ways could I attach them?
We could attach in 6 different spots.
Yeah, there's 6 different linkage spots. Some are not used, right? 1 to 2, 1 to 3, 1 to 4, 1 to 6, 2 to 3, 2 to 6. There are other options that exist out there. Some would be in alpha linkage. Some would be in beta linkages. Okay? And again, they could be D or L, but again, we're going to ignore the L form here because they're almost all exclusively D. Then they could also have been branched, right? Yeah, so ignoring the branch, ignoring the L, that gives us a whole bunch. Now, I actually meant to pull that over. Sorry, there's your number. 1.7 times 10 to the 14. Big number, right? Just type that for now. We'll come back to it. Big number. So am I after the numbers in this? No, what I want you to be writing about is this stuff. How do I link them? And I don't need you to stop. I want you to tell me it's D or L. Commonly what it is. I want you to describe the variation between sugars versus amino acids versus nucleic acids, but specifically what is it that gives me the variation in sugars? And the linkage is, I get 6 options over there, right? That's a big deal. It wasn't the 20, there were 20 common amino acids. But it wasn't just 6 options. The fact that I could do the alpha or beta essentially made it 12 more options, right? So that's why it becomes really complicated. So if I wanted to give you a password and you said you could use 6 letters, and that's all you can use, and you don't want it to be hacked, what options do you want? Do you want to use your numbers? Do you want to use uppercase, lowercase? Do you want to use your full alphabet? How about some alphanumeric symbols? Wouldn't that be nice if you could have all that option for just the 6? Because you don't want to get hacked, right? The body is doing the same thing. It's saying, how complicated can I make it so that I'm not likely to get copied by accident? Does that make sense, what we're talking about with the sugar code? Why do that? Because just like we looked at with your blood type, there is codes all over your cells that you recognize you and you know somebody else is not you. And it's the sugars that are commonly being used to communicate that information. Okay? So that's the complexity of our sugar code. Because this is on the right on, and I know a common mistake, let me also say this. What is this option called? When I link the carboxylate and the amine together, what is that bond called? Some of you, 50%, will tell me peptide bond, and I will not be happy. What do I want you to call it? An amide bond. Okay, fantastic. When you write that, write to yourself a note, say amide bond, because that's what I'm looking for. I don't want you to just say peptide. You're right, it's a peptide bond, but give me more. Be specific. All right. The extracellular matrix must be outside of the cell. It's communicating information one to another, right? It's a barrier that we use. Yeah, there's a lot of options there for it. Glycoconjugates, the information-rich surface a cell shows to its surroundings. What's it preceded by? Glyco meaning sugars. It's your sugar code. Okay? Wow. For cell-cell recognition, cell migration, immune response, wound healing, blood clotting, also trying to understand. There's information. Syndicans, we're going to talk about. Integral membrane proteins, proteoglycans known as syndicans, other integral membrane proteins, receptors known as integrins. We're going to talk about these, used for cell growth, mobility, apoptosis, wound healing, all the things we just talked about. How does it happen? Well, here's some syndicans. There's also something known as a glycogen, which you'll need to know both of those. Do you see the star on that? What did that mean?
It's on the test.
It means it's definitely on the test. Well, what's on the test? Actually, we'll define it in the next couple slides. But you need to know what a syndican is versus a glycogen. Well, a syndican has a protein core, but so too does the glycogen. The syndican has sugars on that protein core. So too does the glycogen. Well, this one's an integral membrane protein, the syndican. Yes, that's true. What's this one? Well, it's linked with inositol anchors. So actually glycophosphate inositol anchor. What does that mean? It means I've got a lipid anchor here, which is a phospholipid, that then it has the phosphate linking all of these sugars, which then the sugars are linked to the lipid, to this protein core, which has decorations on it. So if that material can be shed into the extracellular matrix so that I can then talk to another cell, how would I shed that information? Does it just
Cleave the sugar.
Yeah, cleave off. And do I want to cleave the sugars or do I want to cleave more? The answer is I cleave more. Actually, we use a protease to cut this off.
Wow.
And we use what would I use here? A phospholipase is actually what we use when I'm cutting the lipid phosphate junction, okay? So this is a phospholipase to cut that information off, okay?
Okay.
So that information can then cross. The syndicant itself, single transmembrane, lipid anchored, same things you just looked at, right? Both shed, interact with a variety of receptors, regulating cell growth. Wow. Is there a variety of decorations? Yeah, there's a whole bunch, right? Don't get confused. This is all of what it's trying to present. Okay? Well, let's look at some more. What was the role of that syndicant? Remember, it was regulating cell growth. These are not on the test, but this is helpful to start digging into biology more, especially some of you who've never studied this. Fibroblast growth factor, that's the FGF, okay? Matrix adhesion, cell-cell adhesion, tumor suppression. Lipicans critically involved in the development of morphogenesis.
In developmental morphogenesis?
Yeah, so what do I mean by morphogenesis? What do I mean by
Transition from one cell type to the next.
yeah, how do I decide which cell type I am? So that's involved in that. Okay. I'm going to show you more on this sugar coat that you just looked at, but we're zooming in on the sugar coat. Okay, that decoration. Now, this in gray is my Na, and NS is the one in green. Na was N-acetylated, and NS was highly sulfated. They're just talking about sugars that themselves have acetyl groups or sulfated groups, okay? 4 schemes that we're looking at here. Imagine that I have a sugar sitting on top of this chain that you just looked at on the outside of your cell. Along comes this thing called AT, which is antithrombin in our case, that you're about to look at. And then it brings by factor XA. For those who haven't studied the blood clotting factor, that is what we're just looking at. So by binding the antithrombin to it, the factor XA happens to come in and bind Y. What happened to antithrombin when it bound to this sugar domain?
Goes through a shift, conformational shift.
The charges on this caused it to bind and then change shape. That actually brought up the factor F. This helped prevent clotting. Oh, nice, because I didn't want to clot. If I had done this instead with thrombin, it's actually the same antithrombin bound, and then this also inhibits blood clotting. Nice. So 2 different factors of the clotting cascade being suppressed by my antithrombin, which makes sense. You might be thinking, now I'm getting it. Okay. What about my fibroblast growth factor? It's a dimer, sulfated domain. This actually helps bring the 2 groups together. So this one, we're talking about conformational change. This one, conformational change increased the binding affinity. This one changed the shape. This one over here brings dimers together. This one right here brings what? What's it recruiting? A lipoprotein lipase. Lipases are the ones that cut up lipids. Did you know that a lipid doesn't go across as a phospholipid or as a tag across the membrane? It needs to be cut up. We transport them as fatty acids. There's a specific transporter, but it moves them as fatty acids. That means when I have a tag and it's going actually to be stored in my fat cells, I cut the glycerol off of it and let it float away, where it is turned into fuel, by the way. And then inside the fat cells, I need to remake glycerol, which seems like a waste. Turns out that it can be problematic in some instances of stress. We'll talk about later. But yeah, recruiting lipases, okay? So these are 4 strategies with sugars interacting with proteins, okay? There is your awesome blood clotting cascade, looks cool, lots of green. You don't have to memorize it unless you're going to medical school. Then you get to, but I'm not going to test you on it, but they will. Great. So, does it look complicated?
Yes.
Yes. Top level, you see things that are blocking it, things that are in red are the inhibiting, things that are in green are the ones that are going. Ultimately, what does it do? It causes a cascade reaction where one reacts with another, reacts with another, and causes your whole bloodstream, actually, with your fibrinogen, to cross-link and clot. Okay? Well, we mentioned the fibroblast growth factor. Maybe we should take a peek at that too. We'll talk about tyrosines and why they like to be where they are on the cell membranes a little later. But a tyrosine kinase is super common. We'll use that in our last chapter talking about insulin. This is a dimer, in this case, of 2 tyrosine kinases, which, does anyone know what kinases do? Maddie, you knew?
They facilitate the transfer.
Yeah, they phosphate, right? They use ATP and they add a phosphoryl group to other things, usually marking them. Okay, good. All right, so fibroblast growth factors recruited to this, and then we have a signaling pathway. The JAK-STAT, the ERK pathway, nice. It also binds heparin and heparin sulfate in this case, which you saw up here. Well, let's take a look at where the heparin binds. What was heparin again? The most what? Highly negative biomolecule. And yes, it was also a anticoagulant. Okay, so fibroblast growth factor binding to heparin here. How is it binding? What do you think blue is here? What do you think red is in this picture?
Blue's got to be a positive charge.
Blue must be positively charged amino acids. I kind of would have done the other coloration, but you know, that's just me. I didn't make the figure, I just labeled what they actually are. How would I have known that that must be the case? What are these sulfates? They must be interacting with, because they're negatively charged with positive amino acids, okay? And once in there, they prevent the activation of these fibroblast growth factors. Okay. So it's showing you some interactions with those. Integrins are another one. You guys have studied integrins, or you should have in biology. We're not into a biology lesson in all this. We're really talking about sugars and how are they used. But integrins also interact with these proteoglycan pieces, right? And when they interact with this extracellular matrix, the integrins, which are actually put as that spacing that you're seeing, they're actually 70 angstroms apart when they are activated. An exact spacing that we've studied them ad nauseum. And when they do, they actually communicate from the outside to the inside. There is communication that goes from the inside to the outside across integrins, and it communicates from how do I communicate inside and outside of a cell. The integrins involved in cell-cell adhesion, cell proliferation, signaling, migration, tissue repair. Seems like an important fact. Yeah. Okay.
I do have a question.
Yeah.
So, is the way that information travels through the integrin, is it just the shift from the
dimer,
dimer between the distance between the 2 parts of the dimer?
Yeah, exactly. In their inactive state right here, that conformational change when it's activated, because it's in this case interacting with the sugar that it's known to bind to, it communicates with that specific spacing to the actin filament inside.
So, in other words, it goes through a conformational shift, which shifts the side that's inside the
Placement
cell
on
as
the
well.
actin filament, which actually is,
What does stuff? Cool.
yeah. So, cool, a way to communicate from inside out. There must be other ways other than what we just looked at for a cell to recognize the sugar code that we just talked about. And there is. They're known as lectins. Okay? So a lectin is a protein that recognizes specific carbohydrate sequences. When we talk about tomorrow, the flu, we'll talk about hemagglutinin, as if I were talking about H1N1 in the flu code. Hemagglutinin is the H. What hemagglutinin is two roles. One is it recognizes and binds to a specific site. How does it do that? It's a lectin. And then it actually also acts as a fusion protein, which we will study later. Okay? So, all these things we just talked about are also happening with lectins. They're not antibodies, but essentially they're proteins that are acting like it. They just aren't on the immune side. They're saying, this is the information you're transferring to me. Can I actually use lectins and purify them? Yes. Could I use them to bind specific type of proteins? Absolutely, and we do. Meaning you can buy columns coated with certain lectins that will bind certain proteins that have that specific sugar code on it. That's pretty cool, right? How long had the red blood cells lasted, by the way?
A few hours.
A few hours. Wow.
Nuts.
When the lectins are interacting, it seems to me like a carbohydrate, at least on the surface, looks pretty much the same, right? A few hydroxyls up, down. Obviously, the hydroxyl position matters, but did you know you could put all your hydroxyls on one side of the ring structure and the other side not? Which would make one side what? Polar and the other side? Well, that's clever. Sugars can have a polar, non-polar side? Neat. Okay, so that's good. What about their specific coordination? Here, we're looking at mannose. 6-phosphate. It's got a phosphate group. It's also mannose. It fits exactly into that lectin binding spot and is recognized. Okay. pH dependent. Okay. Here's a few subcategories of lectins known as selectins. Oh man, they should get up on their vocabulary. So a lectin is one that just protein that binds sugars. Selectin is one that's beneath that. And here's a decoration of them. Notice that when we get to our virus binding to a sugar code, which we'll do tomorrow, that makes it a selectin itself, not just a lectin. Cool. All right. Here's your immune response. You thought it was so simple, right? It is. I have this leukocyte. So for those who aren't up on it, when I learned about things clear back in elementary, if I didn't take a biology class, what did I learn about? Red blood cells and white blood cells. The leukocyte, for all intents and purposes, my white blood cell. Okay, coming along to handle my inflammation site, and how is it going to know that it's an inflammation site?
There's selectins that are expressed in that area.
In this instance, we're talking about the selectins, P-selectins, and the glycoproteins that are actually responsive for the integrin itself are going to communicate a message. So first, I'm catching this P-selectin. And as I do, then it actually pulls it closer, and then the integrins communicate, and the integrins communicate to the inside of the cell, not just a docking instance, but talk to the inside of the cell and say, this site or neighboring sites are under attack. We got to go to work. And they do. And then they go in and they'll envelop and destroy, right? This is a beautiful response. What's it driven by? Protein-protein interactions, which are sugars. In fact, were there sugars on this one? Yes. Were there sugars here? Yes. They were. They're like sugars decorating the inside of cells and outside of cells, communicating the information we wanted. So, I'm a mass spectrometrist. That's my job. And actually, I'm a bioanalytical mass spec guy. Meaning, as I have a degree in biochemistry, what's my actual specialty? I measure stuff in living things. That's my expertise. And I do it on not just a few things. I do it on the rainbow of anything you can imagine. But one of the hardest things to measure, the one that's Nobel Prize worthy, is this one. That sugar code we just talked about and its complexity, it's very complicated. And how much, if I'm looking at all hexoses, how much do they all weigh? The same. So what, are they branched? Are they linear? How can I figure this out? So we have ways that we can figure out sequencing, but it's so limited, so very, very limited, that it's not considered solved. We don't know what the information is on most proteins. We just know it's important. Meaning of the 300,000 decorations of sugars and other phosphoryl groups, we can recognize the phosphates really easy, but the sugars are complicated. And so we ignore it. We literally ignore it. It's this protein, that's all I care about. Does it make a difference? Yes. Absolutely. People would argue it has the difference in whether your immune system's going to be attacking it with an autoimmune disease. People would argue that this has everything to do with communicating, for example, speaking of that, if I was type 1 diabetic, what am I attacking? My own insulin? Yeah. So these are issues that we want to solve. And yes, you see some awesome reactions. You don't have to learn any of that, so you probably won't. But we use NMR, we use mass spec, we use reactions, where we're trying to solve the sequence. When you are ready to solve this, you can get a Nobel.
Okay.
If it sounds easy to you, great. Just alone cutting off the single sugars and trying to separate out the phospho sugars with just one phosphate, what do you think those look like on a retention column? I could get less than a second between all of them. Good luck. Meaning it's complicated. It's a lot more complicated than we want to admit, and it's not going to be easy. Okay? Yep. Yep.
Oh, man.
Okay. Yes, we do reactions where we try to model it, we use heavy oxygen. Yeah. Okay. Well, all that looks great. What about extra credit? Does Brother Bowden give extra credit?
That'd be nice.
I'm going to be honest with you. I'd like to give you some extra credit. If you'd like it, I'd like to give it to you. It's a pretty stupid question, but it's one as biochemists, since we're on sugars, that always comes up. As it comes up, I'd like you to dig into the work, into the research, and figure out some questions, okay? Now, I'm going to give you this basic challenge, okay? I'm going to be the company that just hired you, and your job is to make a Twinkie knockoff. So you're going to bake this sponge cake, fill it with cream, sugar, nasty but delicious. And the other, and the other one you're going to do is one that's going to be an acidic drink. Okay, fruit juice with grapefruit, lemon, lime, sweetened to taste good. Artificial sweeteners are the name of the game. That's what you're trying to use. Sugar substitutes, artificial sweeteners. Are they all bad? Are they all good? Does it matter? Is it going to cause diabetes? What is the good? What is the bad? Are they cancer causing? Are they not cancer causing? Is this all a government conspiracy? Is it not? Tell me the truth, okay? What do I actually want?
Okay, handle the truth.
Include the name, structure, sweetness index, and calories. For how many? 4 common sugar substitutes. You get to choose them. 4 common sugar substitutes, okay? Which would you choose for each application and why? Also include any dangers or problems with stability. So if you choose it, if your choice was that, are there any problems with stability? Obviously, don't choose one that high temperature destroys it and then tell me that it's destroyed in high temperature and put it in your baking. Right? If it's going to respond to acid, low pH, then don't put it in your acidic drink, right? All right, so 4, tell me about them. Extra credit, when's it due? Friday night.
How do you want us to submit it?
Online.
Like, the feedback fruits?
Yeah, meaning this. It just showed up.
Oh, wow.
How are you submitting? Yeah, you're going to submit electronically, okay? So if I were looking at like you would, it's got a way to upload an assignment to it, okay? Seem reasonable? Could you answer other questions? While you're there, why not discover the very questions you have? Why? Because I feel like this biochemist, somebody who's taken an upper division biochemistry class, you should be able to answer some questions intelligently about these. Instead of lead on to folklore that may or may not be true. To public opinion, which may not or may not be true. Go to research articles and answer some real questions for yourself. Okay. Yes, these answers will come easy. But why not explore some? See what you learn. Okay? All right. Well, that sounds great. I'm ready to go. Well, did we answer the questions we were after? Huh. Yeah.
Okay.
Okay. So, then in the last 7 minutes, should we watch something that'll help you get up to speed? Let's see if I can get up here really quick. Okay. Okay. I want to show you a clip. It will take you just a couple minutes. This is going to be a lovely little clip. All right. So, for those who haven't taken advanced virology, when you go to read this, this is talking about the flu in the way it attaches to and goes into your cell. Okay? The first thing you should know is that it's going to attach via hemagglutinin to the cell. It's going to release via neuraminidase. So the way into the cell is through hemagglutinin, binds, and goes inside. The way out is through neuraminidase, which actually is cutting off the very same things, which is sialic acids, which you already looked at the structure of. Let's see what this clip tells me. Oops, sorry. Okay.
Containing, if you move in and take a very close look, guy has the flu, could be any flu, and here's a
droplet of the sneeze.
Okay, you want to see
Look,
it.
see, each one of those little purple things is a virus, and there are a lot of viruses floating.
Okay, let's start.
So let's say that this guy has the flu, could be any flu, and here's a droplet from his sneeze containing, if you look at it, take a really close look, see each one of those little.
This has great graphics,
and there are a
which is
lot
why
of
we're talking about it.
floating through the air, some of which go inevitably up this unfortunate man's nose.
How did that guy feel when you ripped off half of his face?
It was interesting because we did it while he was sleeping.
Okay, I am talking with medical illustrator David Belinsky, who designed this video for Xyrus Research Company. So here comes the virus, and it's going to land on one of this guy's throat cells. So notice it's covered with little yellow knobby things that you call.
These are
Those are the keys.
Yeah.
hemagglutinin,
Okay, if the keys on this
sialic
virus happen to
acids,
fit the lungs,
sugars.
which are those little purpley stick-uppy things on the surface of the cell, if there's a match, the cell, watch this.
Lock and key, what does it actually do?
In.
Changes shape, right?
What's this?
This is the welcoming committee. They all interlock with each other and they pull this membrane down into the cell.
And down it goes deeper and deeper, and that welcoming structure disperses, and the virus capsule bursts, and out comes the secret recipe for how to make more viruses, those little noodly things. So this unsuspecting
Our next unit, our next part of this unit,
virus recipe
nucleic acids, right?
into its own command center, the nucleus. Oh, it's so.
And they are immediately recognized by this big pink molecule, which is a mini factory.
Yeah, what is it doing?
It threads the nuclear material, the instruction code of the virus, through one hole, and out another hole comes a brand new instruction set.
So it's a copying machine making copy after copy after copy of virus recipes, which then go out of the nucleus to little chefs, those blue peanut things.
Chef?
They cook up proteins that
Liposome, right?
reassemble into baby viruses, and then out they go. They get covered up and head to
Proteins just covered them. Proteins synthesized by the cell,
Here they come.
released by neuraminidase.
After virus, after virus, after only one virus entered the cell. But how many came out? Well, millions. Millions. So if one virus can produce 1 million babies and do it again and again and again, how come this guy doesn't just drop dead, I don't know, in like 10 minutes?
Well, because you have about 100 trillion cells.
I see. So 1 million viruses is just a drop in the bucket of 100 trillion cells. Anyway, remember, you do have your own immune system, which when it sees a virus, usually...
This is the macrophage. I know that you dreamed of it that way, me too. It was,
With any luck, your
wow.
immune system will work just a little faster. So, yes, viruses, all viruses want to spread, that's what they do. But
One more time.
We can keep them in check.
Gotcha. Okay. So, hopefully, in looking at that, as you read this paper to understand, and it is a little dated, we are trying to understand what is hemagglutinin, what is neuraminidase, and ultimately, how we recognize the difference between somebody like the bird flu that's been going on and you didn't get sick versus why didn't you as a mammal? By the way,
Because they have different keys.
different keys, what are the differences in the keys? Let's examine it. Yes.
So in the written response, I don't, probably not be able to describe it exactly.
Yeah.
Talking about the shed.
Oh, it wasn't Avery, it was this other guy. I thought I sent you the,
Yeah, just a
Purple guy
did you
explodes.
check your should just make sure.
Okay, you guys, see you
All right, I
guys on
can't go.
Wednesday. Welcome back.